Okay, welcome back. Um, it's my great pleasure to introduce kick off our section of talks on the life sciences, David Carey, Associate Chief Research Officer and Director of the Weiss Center for Research in the Geisinger Clinic. His primary research interests are in the area of genomic medicine, vascular biology, and extracellular matrix biology. He's helping lead an ongoing effort called the Geisinger MyCode Project, a large biobank of samples from patients linked to data in their electronic medical records as a platform for genomic medicine research. He's the co-author of more than 100 research papers in high-impact journals, such as Journal of Cell Biology, Behavioral Chemistry, Neuroscience, and Annual Review of Physiology. Thank you.
All right, thank you, and it's a pleasure to be here. I think it's been a really interesting program so far. My talk is going to be a little different. It's going to be less technical than some of the things that you've heard this morning. What that means is I'm not going to show you any equations. I don't know whether you consider that a good thing or a bad thing or not. We can decide that later. What I do want to talk about is I want to give you an idea of some of the opportunity that's out there in the medical world for applying some of the techniques that we've been talking about today. And some of that was alluded to in a couple of the talks this morning. And I want to sort of take up that theme. The other point I want to make is that the ability to have access to and then model large data sets from patient records is a fairly new endeavor. And until recently wasn't feasible because we didn't have the data sets. And that's largely a result of the fact that the medical community has been very slow to move into the use of digital patient records. For those of you that are used to dealing with large data sets, it might come as a little bit of a surprise, but still today in the US, a significant number of patient records are still done on paper. And for example, the medical school here at Rochester is just now they're several years into the implementation of a large electronic medical record program. But that's something that over the coming years is going to become increasingly available. And I guess my plea to those of you who have the skills to study and model those data sets is I think it's going to provide an enormous opportunity. So what I want to do is tell you about Geisinger Health System, which is where I work, and tell you about some of the data assets that we have and how we're using them. And as you heard in the introduction, one of our interests is not only in modeling clinical data, but also in generating and using patient genetic data and combining that with the clinical data. So, there we go. So this slide, I think, encapsulates everything I want to say, and most of my comments are just going to elaborate on some of the details. So we're an integrated healthcare system that gives us great advantage in terms of collecting comprehensive patient data, and I'll tell you a little bit about what that means. This really is also made possible because of a very non-transitory and supportive patient population, and that really has been critical to creation of this central biobank, the MyCode project that you heard about. We collect patient DNA and blood samples. Geisinger was an early adopter of electronic medical records, and I'll give you a little bit more detail on that in just a minute. And our ability to have these records and these samples and then link them to do discovery and validation research is really, I think, a very powerful platform. But also we have the ability to take that information back to patient records to close that loop and make sure that it doesn't stay as just a discovery, but as a way to continuously improve the patient care process. So we're an integrated system. What does that mean? Well, I'm not going to go into all the details in this slide, but we have Geisinger Clinic, which is a giant physician group practice. We have 1,100 physicians. They all work for the same boss, both primary care and specialty care, and a lot of advanced practitioners. We're a teaching hospital, so we have about 400 residents and fellows. We own all the facilities where these providers provide care. That includes 6 hospitals. 2 of those are large tertiary quaternary teaching hospitals. We have 78 primary and specialty care sites, ambulatory surgery centers. So all of that is under one roof. We also have our own insurance company. We don't insure all the patients we provide care for, but a substantial fraction of them we do. So but all of those separate entities, which in most cases would be separate businesses, are all roll up together under what we call the Geisinger Health System and report to a single president and chief executive officer, Glenn Steele. To give an idea of our geographic footprint, we're located in north central and northeastern Pennsylvania. Of course, Philadelphia is down here, Pittsburgh is here. Our patient footprint covers most of the rest of the state. So I'm not going to go into all the details here, but you can see each of these dots or crosses or squares is a different care site that we have scattered throughout a 31-county region within that part of Pennsylvania. And we go up to the New York border, to the New Jersey border on this side, and cover most of the rest. So it's a large geographic footprint. About 3 million people live in that area. We're the largest healthcare provider, which gives us, I think, a great market advantage in terms of capturing both primary care and specialty care data. So in order to mine data, you have to have data. And again, I mentioned we were an early adopter of electronic medical records. So where many health systems are now struggling with just the implementation of these infrastructure, we did this and finished this more than 10 years ago. So we've had plenty of time now to accrue longitudinal data on these patients. And now we're working on methods for extracting that data and modeling that data. And I'll give you just a couple of simple examples as we go along. So our decision, we use Epic as our electronic health record system. It's one of the major systems in the country. The decision to implement Epic at Geisinger was made in 1995, so well ahead of where most of the rest of the country did it, which has been a real strategic advantage for us. It's fully integrated now across all care sites. We have a web-based patient portal that we can communicate in a secure manner with patients. So to get an idea of sort of the size of the data set, there's more than 13 million individual patient encounters that are recorded in the electronic medical record since 2004, and 1.8 million in 2012, and that number has been going steadily up. So there are about 740,000 patients unique patients that have had an encounter during that period. So it's a very large data set on a large, very stable patient population. We think is a great asset for data mining. So in terms of there we go. No, we did this. In terms of the sort of data elements that are recorded, it's pretty much everything that happens in a patient encounter. Inpatient, outpatient diagnoses, surgeries, emergency department data. All orders are now done by computer order entry, electronic prescribing, all labs, procedures, diagnoses, scheduling, demographic data, imaging, et cetera. So it's a fully electronic data set. Now, one of the things about Epic, it's a transactional EMR tool. It's not a good database. So we've created a enterprise system-wide data warehouse. We call it the Clinical Decision Intelligence System, or CDIS. And that's usually the place that we would go to for this data. So a lot of the data in there is generated from the electronic health record itself. So Epic has a reporting tool called Clarity. That's the way we get most of the data out of that record. But we also combine insurance claim data from other databases, billing from other databases. Our pathology department, we have a central system-wide pathology department. They use a different database called CoPath. It talks to Epic, but all the data from that database goes into the data warehouse. And there are a host of other sort of standalone databases, many of them tied to particular medical specialties. So one that's used for recording pulmonary function data is called Breeze Suite. There are others for cardiology data. So that's just an example, and tumor registry data. So this is kind of one-stop shopping for all the data that we have. So one of the things we've done is use this data to identify patients with particular traits of interest. And so we call those phenotypes. And so the basic way we do this is you come up with an idea of a particular patient you want to identify. So sometimes this is driven by a research idea. Sometimes it's driven by the desire to manage a particular group of patients in our population. So for example, can we identify all patients with diabetes, or all patients with chronic kidney disease or heart failure? Then what we do is work with content experts, usually these are clinicians, and sort of talk to them about what are the diagnostic or procedure codes, lab values, other data that we would have that would be useful for identifying patients with those traits. We'll then do an initial, this is an iterative process. So we'll do an initial query of usually the data warehouse to find out, sort of give a rough idea of what we're dealing with, and then get down to really developing a more complex phenotype algorithm. And you heard this morning some examples of how we do that using this type of data. And sometimes we're looking for identifying patients, if it's a research study, cases or controls, or other ways to manage individual patients. And then we'll execute the algorithm against the data, determine how well it works, and then go back and refine that algorithm if it needs further refinement. And then if it's an important research study or some other variable for which we need really highly curated data, we'll actually sample or take a random sample of, say, cases and controls identified with the algorithm, do an actual chart review, electronic chart review with a content expert, and then we can calculate the positive and negative predictive value. And we've set a fairly high bar, especially for research studies, in terms of being able to do that. And in many cases, we can get to PPV and NPV of better than 95%, sometimes approaching 100%. So we're also interested in combining this patient-level data with genetic data. So I mentioned the biobank, and I'll say a little bit more about that in a minute. But so we use our clinical data to create these validated phenotypes. From our patients, we collect blood and DNA samples that we use for research, and we're generating genomic data. And one of the things we want to do is to combine that to look for new gene disease phenotype associations. So our biobank is called the Geisinger MyCode Project. This is an informational brochure. When the patients agree to participate, so they're approached during an outpatient encounter when they come to one of our clinics. We recruit from both primary and specialty clinics. The eligibility list is very broad. Basically, if you're a Geisinger patient and you can understand the consent form, you qualify. We even enroll children now, so it's a great source. The patients, when they do this, they sign a consent form that so that they volunteer, opt in for this. It allows broad research use, so it does the consent does not limit the use of these samples to studying a particular disease or condition. One of the beauties of having this advanced electronic medical record system is when a patient consents, that information is entered into their electronic health record as an encounter, and that triggers an automatic order to get a research blood sample the next time they go to the phlebotomist, to the clinical phlebotomy clinic, to get a blood sample that their doctor orders for medical testing. So it's a really slick way to do this in a way that's very efficient and really adds very little extra effort or expense to creating this resource. Also, because the samples are collected through the clinical infrastructure, they're under what's called a CLIA umbrella, which means that they can be used for clinical testing. We can get serial blood samples. And because it's such a streamlined, high-efficient process, we get very high consent rates. So to give you an idea, we have about 45,000 patients that have consented to give samples. It gives you an idea of the breakdown of sort of clinics from which they were enrolled. We're actually getting ready to gear up to substantially accelerate the rate of patient enrollment. So our goal is over the next 5 years to get up to about 150,000 of our patients that are enrolled in this program. So we get blood and DNA samples, so we've been starting to generate genomic data from these samples. And I'll give you just sort of a partial list of some of the data we're generating. So about 3,000 samples have been used for analysis using something called the Illumina OmniExpress array. So it interrogates about 700,000 single nucleotide variants in the genome. That data has been imputed to something called the 1,000 genomes, which is a large human genome variant data set. Imputation then gives us about 12 million variants on each of these patients. So again, we're getting up to very large data sets. A second type of array that we've used is called the exome array. This interrogates coding variants in the genomic, in the gene coding portions of the genome. It's about 250,000 variants per individual. And then we just finished another almost 10,000 patient data set with the newest version of these arrays, which has about 500,000 genetic variants. So we now have almost 18,000 patients for which we have this extensive database. So again, thousands and thousands, hundreds of thousands of data points on now tens of thousands of patients. So we can combine these, and what we've done One of the things that this slide explains is how we pull all these things together. So most of the data that we collect comes from the electronic health record, EPIC, that reports into CETUS, our data warehouse. It pulls in data from other sources. Now, because this was collected as part of clinical care, there are federal laws that protect the privacy of that information. And so this so-called protected health information is sort of behind a firewall. And it's unless it's used for clinical treatment, payment, or operations, there have to be special dispensations by which you can access that data. So what we have developed at Geisinger is a process we call the data broker process. And there's under that, a certain group of data analysts have permission to go into that data, pull out the data elements that are needed to do the types of studies that I described, say create a phenotype algorithm. And if necessary, strip that of the personal identifiers and then give it to the investigators who are going to use that data in a way that's compliant and protects the confidentiality. We have a similar process for the genomic samples and the data. The biobank and the data associated with that are kept in a separate database, but they also are managed by the same data broker process. And the data brokers can link the clinical data to the genomic data. And one of the beauties of this is that the data brokers maintain the link of the data to the patient, so we can longitudinally refresh this data as we get more information on these individuals over time. So just as a proof of concept, we did 3 simple genetic association tests using known genetic variants that have been previously shown to be associated with important clinical phenotypes. So we picked 3, 2 different variants on chromosome 9 that are associated with cardiovascular disease, 2 variants in this gene that are associated with type 2 diabetes, and 2 different gene variants that have been associated with obesity. And using the existing genomic data and only electronically derived phenotypes using the methodology I explained, and this gives you an idea of some of the sample sizes of the individual patients that were identified or used for these studies, we're able to replicate these genetic associations with very high degree of statistical significance. I'm going to now give a couple of other examples, and in this slide I put up to illustrate the value of having longitudinal data. So we've done a number of studies that target an adult obesity clinic that we have. Obesity is a big health problem for us where we live, and as you know, it has a lot of comorbid medical conditions like diabetes. We have a large clinical program for gastric bypass surgery where many of these patients with severe obesity are treated, and a lot of them have diabetes. And one of the, there are really 2 advantages to this procedure. Everybody loses weight, and in patients that are diabetic, and about 40% of these patients have diabetes at the time of the surgery, most of them, their diabetes symptoms go away. So that's one of the reasons why this procedure is becoming more popular clinically. But one of the things we wanted to do was do long-term follow-up studies on patients after they've had this procedure. And so in order to do that, again, we have to have data. So what this chart shows is so these patients all come back to the same clinic, one of our clinics on the Geisinger Medical Center campus. And so what this shows is the percent of patients that come back for their follow-up visits over time after surgery. So this is 48 months or 4 years. So more than 80% of these patients we're still seeing and collecting data on 4 years later. And if you're not familiar with sort of the usual fragmentation of healthcare in the US, that's a really extraordinary degree of follow-up. And again, it gives us a great advantage in terms of having complete data sets and longitudinal data sets on these patients. I'm going to skip that one. So one of the studies we've done is to look at genetic variants that are associated with clinical outcomes after surgery. And 2 outcomes that we're particularly interested in are long-term weight loss. Everybody loses weight after surgery, but unfortunately, beginning about 6 to 12 months after the surgery, many patients stop losing weight and regain weight. So it'd be nice to know in advance if we could predict who is going to be a success and who is going to be a failure. The other thing is not every patient with diabetes who has the procedure loses their diabetes symptoms. And again, it would be nice to know from a population health management who is going to benefit from this procedure and who's not going to benefit. So one of the things we did, I'm just going to show you one set of data. So there's a gene called the melanocortin-4 receptor. It encodes a protein that's expressed in your brain in the hypothalamus, and it controls eating behavior. And we know that from mouse studies and other studies, including in humans. So we looked at sequence data. So we sequenced the entire coding region of this gene in about 1,400 of these Geisinger bariatric surgery patients and about 450 lean controls. And then this is just for the molecular biologists in the audience, this is a structure of the gene product. It's a membrane protein that generates an intracellular signal that controls eating behavior. So it turns out in this obese cohort, almost 6% had a non-coding variant in the gene. Some of them were rare. In fact, we found some that had never been seen before. But almost 2% had a particular variant in residue 251, where an isoleucine was changed to a leucine. And and that hadn't been really reported before in terms, I mean, it had been reported, but the functional significance of that wasn't known. So that's the position of that single amino acid change. So we were able to go back to our data and ask, did that make a difference in the patient outcomes? And the answer was, it did. So if we look at, again, this is patient weight loss data. So this is time 0 is the time of surgery. The black circles are patients that did not have that genetic variant. You see they all lost weight and then they sort of stabilized here. This is aggregated data from over 1,000 patients. But the ones that had the variant actually were substantially and significantly better performers long term. And the difference in weight here was about 9 kilograms, which is 20 pounds. So that's a substantial difference in long-term weight loss. This is also a measure of insulin resistance, which is a way to measure diabetes. These individuals who had this variant also had better diabetes symptoms resolution. So again, very important clinical findings on these patients that we're now following up. And the doctors who take care of these patients are very interested in using this and other types of data to really manage these patients from a long-term healthcare perspective. We're part of something called the Emerge Network. I'm running out of time, so if you're interested, you can look this up online. It's a network of 9 sites. The funding comes from the NIH, but it's basically places that have similar kinds of assets that are sort of working together to develop these kind of approaches. And I'm just in the interest of time. So to give you an idea of some of the phenotypes that we've been working on through the Emerge Network, it's a pretty broad list. So it's everything from aortic aneurysms, venous thromboembolism, ocular hypertension, asthma, cardiorespiratory kidney disease, atrial fibrillation, so you can read the list. So it's a growing list of phenotypes that we're now studying using these kind of methodologies. So I'll put this up again. This is where I started. I think the message that I'd like you to take home is that places like Geisinger and other integrated health systems are great platforms for generating large volumes of patient data. Now that we're moving into an electronic world in medicine, I think there's enormous opportunity to model this data to really, as one of the speakers this morning mentioned, we're paying way too much for healthcare and not getting, the value for that dollar. So this is one of the approaches that I think we can take as a nation to really use this data to manage our health better. Thank you for your attention. Questions?
We have a couple questions while the next speaker sets up. Is Alex here?
He's here. I saw him. Oh, there he is. Okay.
The data that you share onto the Emerge network,
Yes.
are they only of patients who have consented, or how do you take care of the privacy?
Yeah. So good question. So yes, those are all patients who've consented, and we only send de-identified data. So that data is stripped of the personal identifiers.
That's not usually sufficient. You could still have people identified based on data.
Well, so there's a difference between identifiable and identifiable under the HIPAA regulations. So, did that answer your question? And I think there's a big debate now in the country about the sharing of genetic data, because it's also obviously a unique data set, and with the right reference information, you could use that to identify an individual also. So, but it's not, that is not currently one of the HIPAA identifiable elements. So,
So Dave, I was curious about when you have information about individuals genetically who will do better from bariatric surgery versus worse, how does that play out on the insurance product side of your business?
yeah, so another good question. So currently there's a federal law called GINA, the Genetic Non-Discrimination Act, that we think would preclude insurance companies from using that information to deny coverage. But so for this research, we keep this at arm's length from the insurance company. So, but we're actually now at the point of, in fact, we just amended our IRB protocol around this because we're getting to the point where we're getting data that we think will help patients. So I think there's an ethical obligation on us as a health provider to share that information when we think there's a good case that they need to know it. So. Yes. And, you know, you could argue that maybe that's a good thing, because you could then prevent them from going through a procedure that you know they're going to fail, or try an alternative procedure.
How do you envision implementing the personalized medicine aspects of this? So, for example, with the bariatric surgery and the gene predictor, do you envision developing a CLIA-compliant specific test for that gene variant? Or do you envision whole exome sequencing all patients when they come through the door and then using that information?
So eventually, I think we'll have either whole exome or whole genome sequencing data on everybody. It's coming now. We're again embarking on a new project to do whole exome sequencing on tens of thousands of our patients. It's a research study, but we're also going to find things that are clinically useful. Through the Emerge Network, we're doing another study where it's a smaller subset of patients, it's about 1,000 patients, but we're specifically targeting genes that we know are involved in drug metabolism. So there's sufficient evidence for some of those that there's information there that could be useful to a physician when they're prescribing medications, either to avoid an adverse event or to choose a drug that would work better in that individual patient. So in that case, there's three drugs that we're going to be targeting for that, and a subset of genes in this panel that we're looking at, that we're developing methodology to actually return that to the patient's medical record.
Okay, thank you very much again.